Hello, Med13L community. We're going to try something new today, which is a video interview. I'm very excited to have with us Dr. Rachel Heilmann. For those of you who have been following our newsletters, you'll know that Rachel is the first full-time dedicated person that we have brought on to the Med13L Foundation. Up to date, we've been able to accomplish everything we have with an all-volunteer organization, and we are super excited to have Rachel. We also got to know her for about a year on a consultancy basis, and so Rachel had a very big impact on the organization before she even joined. And so without further ado, I would like to introduce everybody to Rachel. So Rachel, maybe if you could begin by describing your background to the community and then how you came to work with Med13L.
Yeah, thanks, Nick. And so you alluded to it a little bit. My background is actually a little bit more non-traditional than maybe some other folks who lead research and development in other organizations, other small nonprofits. My background's in clinical pharmacy. So I have a doctorate of pharmacy and then have about 14 years of practice in a large healthcare integrated system. And managed teams and medical specialties and was fairly heavily involved with specialty medications and coordinating care even with our children's hospital here in Denver, Colorado. I do not have a child with Med13L related neurodevelopmental disorder, but I am a rare parent. So I had a daughter in 2019 who subsequently passed away from her ultra rare disease, and founded my own nonprofit to really be on the same journey as the foundation, Med13L Foundation is, and ran into Terry Jo Bichelle and Combined Brain, and she I guess was semi-impressed with my knowledge of my own rare disease that I was talking about, and offered to take me on in a fellowship capacity. So I really spent the last year learning with Combined Brain, and the Med13L Foundation rented my brain. And so I joke and say that now they own my brain. And I've had the pleasure to get to know Nick and Vanessa and Katie and Kelly and Kim and Lacey and other members of the community really in the last year, and developing the strategic road plan and participating in biorepository collections and meeting many members of your community already, and your kids, which has been great. And if you don't recognize me, you may recognize me with the Mario hat on. Because that is how I have shown up to blood collections across the country, always in costume to bring smiles to kids. And
Yeah.
I don't take myself too seriously when it comes to that, but certainly am aligned with the direction of the foundation, and have enjoyed working and putting a lot of stuff in place for them for the last year, and hope to for many years to come.
Fantastic. Well, you obviously have a fantastic background for this role, and I can say firsthand, I have two children who do not particularly like getting blood drawn, and you have an amazing way with kids, which is obviously a huge boon for this community, including your Mario hat. So we were very, very fortunate to connect with Combined Brain. We talked about them before. They're really a fantastic organization. And had a great experience working with Rachel. We were very sad when we learned that Rachel had to go on to her next kind of next stage of her career. But ultimately had the idea of bringing Rachel on for time with Med13L, and we're very grateful to have been able to do so. One of the things that you worked on with us, in that Combined Brain rent-a-brain capacity, was the strategic plan, which really helped give us a roadmap of where to go and how to prioritize things. Could you maybe give a brief overview to the community of sort of the output of that strategic plan?
Yep. I'm just going to share my screen here, and hopefully this will work in a capacity that people can read and understand. And so this is, gosh, an update that's been just over a year in the making. I presented the original deck in February to an international group of researchers and clinicians and board members and colleagues from the Med13L Association in France as well. And so I've been kind of cobbling and keeping this, trying to keep this up to date over the last year. So let's see here, let me get this. There it is. So this screen, and I know there's a lot going on here, but this slide really maps, the resources and tools that it takes end to end to really get, therapeutics to the Med13L community. And so when I came on with the foundation in last October of 22, this was part of my job, was really assembling and understanding the resources and the work that, you all have done in collaboration, for the last few years. And so what you'll see here is the things, the text that's highlighted in green really means that you've met that, you're really moving forward, that those tools and resources are already in flight, relatively complete, and really more in a sustaining mode, and with some additional sort of things that need to happen to keep it there. Purple is in process, so we're starting at the very beginning, working towards turning them green, and red continue to be areas of opportunity that we'll continue to work towards as this toolkit continues to be expanded. So the things that I really want to highlight, and things that I've been able to work really closely with the board in the last year on, is really around the antibody development, which is actually green. So the Med13L Foundation funded antibodies, which antibodies essentially can be used to recognize the Med13L protein. And we found through talking with researchers over the last year that this was really lacking, and there wasn't a great one that was available that was consistently providing the right information and reliable information. So this has been developed and tested. Preliminary results are expected to be coming actually here in the next week or two, with sort of the validation of and results of what those have shown. And this can really add to almost anything here in sort of the modeling or cell-based experimental phase, because it's really a necessity to be able to understand and recognize what the protein is doing in the body. This was funded through a company called Pacific Bio, and then the foundation additionally funded Caddy Kang at Vanderbilt University to really test and validate those. Caddy is also working through some particular drug screens and testing some separate drugs than what Rarebase has done, and that many of you have participated in the last year. The other thing I want to highlight on this page is really on the mitochondrial rescue studies, which is underway, and I'll talk a little bit about that here on the next slide. And then this central repository, which many of you have participated in the last year, and these are foundation-owned samples that your children have provided to us to help facilitate this research. And this is ongoing, and we're continuing to send out kits, but there is a very solid lines for iPSC cells, which make neurons, plasma, which we're using for biomarker studies to really tell what's going on in the body on a cellular level, and then fibroblasts, which are skin samples, which can be grown into multiple different types of cells as well. So this has been a huge lift, and a huge thanks to so many of you out in the community that have participated in, and for your kids for participating in providing samples. And then we do have some cool things that are coming up this year. And before I go there, I just need to comment on the ICD-10 code, and I've already memorized it, Q87.85. So make sure you tattoo that on all of your children with Med13L. So this is an ICD-10 code, which is huge, and took a Herculean effort from the board to put together, and in collaboration with Wendy Chung, really go before the CDC CDC and make the case to be issued this. This really goes into your children's medical records, and it helps us know how many kids with Med13L are out there, and we're able to collect that information. It can make it easier for payment and coverage, and it's massive. And there's so many other rare diseases that do not have this. So this is something that puts Med13L, a really long ways ahead of other organizations and foundations, and just a huge kudos to you. I had nothing to do with this. This was already done. I'm just going to talk about it because it's so important that everybody knows Q87.85. And then the things we're really starting to get talking about here, just as I've started with the group in official capacity, is working on international sites. So Dr. Jamal Ghamid, we just found out, got some funding. I've been in collaboration and discussing with Jen Bain, a site here in the US, to really start thinking about a longitudinal natural history study. Conceptual model, which is really hearing from you guys as caregivers, what's important, what matters, what your journey has been like with your child. That's going to take off this summer. Katie Boychuk and I will be heading that up, along with a genetic counseling school student from Rutgers, and then a Gene Reviews article, which is really an informational publication for Med13L that serves as sort of a comprehensive resource for clinicians at first pass. So it's very easy to find, it's very easy to read and understand, and really can help guide care and information at your tip of fingers, which is really important for rare diseases and Med13L. So these are all things sort of in the hopper and kind of coming here in the next year or so. All right, just a real high summary of everything that's kind of been going on in the last year. I'm going to try to keep this short and sweet, but there's so much happening. So a lot of this stuff started even before my time, but I've just kind of kept up with it and been cataloging it and where it is on its process. So Rare Base, which I think most of you are familiar with and participated in, is a drug repurposing study. We're right now in a phase 2 expansion testing library. There continues to be ongoing meetings about that data and that information. So I think that'll just be an ongoing conversation and dialogue that we'll have with you all in the community. SomaLogic, which is a company that does proteomics, which is a biomarker. It's essentially to see what's happening in the bodies of our kids with Med13L, and we compare them to siblings, so it helps us tell similarities and differences. This has already been funded as well, and is in process. And I have been outreaching many of you, or you've been hearing from Kelly or Katie or Vanessa about getting additional samples that will really help bolster those results and give us a little bit more insight. So if you're interested in participating, please outreach any member of the board or myself. Katty Kang, I just talked about that. She's a lovely researcher over at Vanderbilt. She's working through antibody validation, and she's also doing some small molecule testing. Again, preliminary results I'm expecting here in the next couple of weeks. The Randy Strick lab. So this is a really hot topic that we're seeing right now, is this mitochondrial function or dysfunction. And so the Med13L Foundation funded him last year, and his postdoc, Alicia Campbell, who I think many of you have also met or heard of. And so there's some ongoing testing that's happening there just to try to find some consistency in terms of what's happening cellularly in terms of the mitochondria and how it's functioning. But we do have some results that is showing some mitochondrial dysfunction. We're just trying to they're trying to get a little bit better grasp on what that looks like, and then do some small molecule testing to rescue that. So again, lots of sort of drug repurposing things going on or planned for 2024, which is really exciting. The Gruder lab, or Chad Gruder at University of Iowa, who's actually been also working very closely with Randy and Katty, is your designated mouse man which everybody needs in the rare disease space. And Chad has been, really working on developing about a half a dozen different types of mice with different Med13L tissue specific variants. And so this is a really cool way to understand what's happening in different body tissues with Med13L. And then based on this, he will also go down some small molecule testing or drug repurposing. This just got funded. The ink just dried like yesterday. So he's ready to go. He already actually has the mice there. So they're ready to start testing here and phenotyping in the next month or so. Jackson Labs, also mice. And then the big thing on this page that ICD-10 code, we are partnering. I'm just going to highlight real quick the Brain Gene Registry. This is a new partnership that really looks at helping to understand our variants of unknown significance, which is very, very important for you guys so that you have a clear answer for diagnoses, but also because it helps to build and understand the repository behind the different variants that are occurring on the Med13L gene. So this is a really cool and exciting opportunity here for partnership. You all know Simon's, and we're hoping here in the next few months to actually launch with RareX, which is a data collection platform that will expand on some of the base foundational work that you've done with Simon's and really target in on some systems that we're able to analyze and put out information in an efficient and collated capacity. So I know that's a lot, and we probably went way over, Nick, but you guys are like just chugging along here, and I'm just kind of happy to be part of the ride and pull on the horn every once in a while. But so a huge kudos to the board and the community for stepping up and showing up to really fuel a lot of these things forward.
That was a great overview. We have a ton going on. I mean, actually dozens of projects. I'd say Rachel is more than along for the ride. She's leading a herd of activities, and I would say a couple things. Rachel, if you wouldn't mind pulling up that first slide from the strategic.
Yeah.
For a sec.
Yep. Let me share you again there. Oops.
So, one of the interesting things about this slide is, again, it really provided a roadmap. This is from, I want to say February of this year. And
yes, and I've been updating the colors as the years go on.
So there was a lot of red here, a lot more red here, when this slide first came up in February. So we've been turning a lot of things green, and purple in process. And then Rachel really helped us think about the categories of things to focus on. So because we're in early stage disease, we recently sort of identified there are a lot of stepping stones to put in place so that we can find medicines. But Rachel, if you would go to the next slide, please. I want to flag that not only are we putting in place the stepping stones, like the tools to, for researchers to make sure they have what they need to study Med13L, or really digging into the biology to make sure that we understand how Med13L works. But we're actually, you can see here, there are actually 4 projects where we're already looking to test small molecule drugs. That includes Gruder's lab and Strich's lab and McCang lab at Vanderbilt, and then with RareBase. So never any guarantees, and road is sometimes longer than we hope, but we are actively looking to try to find medicines, even in this early stage of where we are. So super grateful for your leadership, Rachel. We couldn't come close to doing all the things that we're doing without you. And I think with that, I just want to thank everybody, those of you who are still watching. There was a lot that we threw at you, but again, it's just a small piece of an even larger puzzle. There's a ton going on. Thank you all for your engagement in research projects, in family meetings, your fundraising support, your volunteer support. We couldn't do this without engagement of the broader community. So thank you, Rachel. Thank you, everybody. I think with that,
Yep.
over and out.
Thank you.